Hey, what's up, everybody, and welcome back to the Sanctuary. I'm your host, Professor C, and we'll be doing some more A&P today. Specifically, we're going to look at muscle structure, deep muscle structure, the sarcomere, and what's called a muscle triad. So, let's check it out. Right, the architecture of muscle can turn out to be a pretty tricky topic, so I'm going to try to be very specific here as I mention these terms and take you through the anatomy of a muscle. And as you can see, the muscle looks quite different than the structure of a bone. What I see are bundles of tissue with wrappings wrapped around them that are made of connective tissue. So I'm going to tell the story here going from big to small, and then we'll dive down even smaller when we have the chance. So let's start it off with saying that connected to a bone is a tendon. And I can write it T-E-N-D-O-N, or a tendon. A tendon connects a muscle to a bone. Remember, a ligament connects bone to a bone, and an aponeurosis, which we will see in future talks, connects muscle to muscle. So if you go out from that tendon, as it gets to the actual muscle, the big chunk of it that we actually call a muscle, you can see that it expands to a sheet of connective tissue, and this is going to be called epimysium. So let me put epi, just to show you this is what I'm talking about. So this wrapping, this kind of sheet, looks almost like a plastic wrap over some food. It's called an epimysium. Mes is the root, one of the roots, myo also, for muscle. It actually means mouse. And think about the old days when you would bulge one of your muscles in your arm. It kind of looks like a mouse is running underneath your skin. And that's exactly where the root comes from. So epi on top of, epimysium is the wrapping on top of the entire muscle. So if I change my ink to green, I can continue the story. Notice here's a bundle, and here's a bundle, and here's a bundle of other things. Now what these are, and I'm going to put an F in here for fascicle, fascicle, fascicle. Here's another fascicle right here. And you see the word right there, fascicle. So the muscle is made of little bundles called fascicles. Now each one of those fascicles, and you can see the white here and here and here, all these white patterns that are running in between the fascicles, those are called perimysium. Peri means around, but just remember it goes around the fascicle, perimysium. Right here, I'm going to write peri in green. There's the perimysium wrapping around a single fascicle here that's been pulled out from the rest of the muscle. And you can clearly see that perimysium wrapping around it. But it doesn't stop there, does it? So if you look even deeper, and I'll go to an orange color here, you'll notice that fascicle, which again is this thing. This is the fascicle wrapped with perimysium. Right here is what's called a muscle fiber, or a muscle cell, and this is a good time to just make the point. A muscle fiber is a muscle cell. Here's a cell, here's a cell, here's a cell. Okay, and then here, of course, is a cell as well. And we've seen this type of muscle before, skeletal muscle here. These elongated, striated, you can even start to see the striations there, multinucleate cells. Now to me, this fiber looks like a pack of circular pack of cigarettes, right? So what you're looking at is the end of a cell, and notice that cell is made of little bundles too, but this wrapping that's around the cell, each cell, is called the endomysium. That's what it says right here. The endomysium wraps each fiber or cell. So the epimysium wraps the muscle. The muscle is made of bundles called fascicles. Each fascicle is wrapped in perimysium. But the fascicle itself is made of bundles of fibers or muscle cells, and each individual muscle cell is wrapped in endomysium. Now, if we could see, which we can't in this image, and it is unfortunate, that right underneath the endomysium, and it's hard to draw it in here, I'll try to do it in red there would be another structure, and that would be the actual cell membrane of the muscle fiber called a sarcolemma. So it's telling us the sarcolemma lies deep to the endomysium. And in some pictures, it's very hard to tell whether they're pointing at the endomysium or the sarcolemma. So I'm going to have to label that on everything so we are very sure what we're seeing. Now notice also, if I change it over to another color, I can kind of take one of these cigarettes or these pencils inside this pack of cigarettes or whatever, and I can pull it out even further, right? And we're going to see this on the next page better, but I'm just going to go ahead and tell you this is called a again, there's that root for muscle, fibril. So myofibrils make up each fiber, right? The fiber is made of little bundles called myofibrils. Now, the myofibril is not wrapped. There is no wrapping around it. So the wrappings stop at the fiber level with the endomysium. But the myofibril is made of two other things down here, and we're going to see those because that's too small for us to see. We're going to call these actin and myosin usually, and there's another protein we'll mention too. Those are called microfilaments. And that's at the tiniest level of the muscle, down where the actual contraction of the muscle will begin. Okay, so the concept we're introducing here is the concept of the sarcomere, but let's dive into it a little bit before we get there. So just remember, this entire structure we're looking at here on the screen is a muscle fiber, which is the exact same thing as a muscle cell. Remember what we saw at the end of the last talk on the introduction. We should see elongated cells, and that is that's not even the whole thing, right? It's cut off. So elongated, striated, you notice these striations here. We have like kind of dark light, dark light, dark light pattern. And then we have these purple jelly beans that are, of course, are the nuclei, so it's multinucleate. So we have all of our conditions satisfied for a muscle cell, elongated, striated, and multinucleate. Now, what is the wrapping here? Now here's where it becomes arguable. Are we looking at endomysium, which wraps each fiber, or are we looking at the sarcolemma, which is the cell membrane? For this picture, this is actually the sarcolemma. So let me go ahead and draw that in here. Sarcolemma, the cell membrane. Peeling it back, we can see each of these individual little circles that make up the fiber, these cigarettes that I sort of talked about in this circular pack. And remember, this is called a myofibril right there. And if we look here in between this distance, we can also see one here, this distance, and another one here, this repeating pattern. These are called sarcomeres. So the concept is these dark lines here are going to be called Z-lines. There's a Z-line, there's a Z-line, any of these dark lines here. Now, you can't see them when you see these patterns over here, but we're going to get even deeper here in a moment, and we're going to get very, very close to a sarcomere. It is the contractile unit, the sarcomere. It is where the muscle will shorten. So all those Z-lines, you can imagine them kind of scrunching up together and making that myofibril shorten, which will make the fiber shorten, which will make the fascicle shorten, which would make the entire muscle shorten, at least to some degree. Now we will be talking about, it says 2 here, but those are the two main players. Those are the ones you always hear about on exams. Actin and myosin, two big giant proteins involved with muscle contraction. There will be a third one we'll see just briefly, but these are the two overlapping proteins that give this light dark pattern. Okay, so here is actin. It's one of those 3 important proteins here, but 2 of them, actin and myosin, are the most important. This one is called the thin microfilament. Sometimes that's all it's referred to as, the thin filament. It's made up of kind of a double helix, a twisted double strand of protein, right? And then what I'm going to show you is, I'm going to have to draw it in here. Let's draw one right here. You can see there's a little circle that I'm drawing right here. I'm going to draw another one right here. Pretend there's a little circle under there. And you can draw another one, say, right over here. Say there's a circle underneath here. It looks like there's something covered by that yellow ribbon. All right, so what we want to picture is something that's going to stick in those notches. So imagine some things hanging down. It looks like they want to fit in those circles we've drawn, but right now they can't fit in because they're being blocked by the ribbon. So myosin is this right here. Now we're going to show a better picture of that in a minute. Right now, just picture it as a long protein with these heads that hang down from it, almost like, again, like a golf club head. Now, it's saying here, those are myosin binding sites, those circles we drew, and myosin's trying to bind with it, but tropomyosin, that's the ribbon, is blocking those binding sites. So right now, these two proteins, I'll just go ahead and call this myosin, so we don't have to look at that arrow, is trying to hook up with actin, but it can't right now because the ribbon is in the way. Now there's another portion of the actin called troponin, and it looks like these three balls right here. Now troponin, if it gets stimulated by some calcium, that's supposed to be a 2+ right there. It says there in the text, when troponin is stimulated by calcium, it takes this ribbon and it shifts it out of the way. And this one would get stimulated by calcium, and it would shift this ribbon out of the way. And when the ribbon is shifted out of the way, it exposes those binding sites. So there's the first picture you're supposed to get of this. We'll put it all together later in a more formal talk. But this is going to be called a sliding filament theory, where actin slides over myosin. Now, let's talk about myosin for a minute. It is called the thick filament. If actin is the thin, myosin is the thick. And I said it kind of looked like a bundle of golf clubs. And this is a better picture of a lot of myosin proteins kind of bound up in a bundle with their golf club heads. You have to imagine these heads being able to ratchet, right? Kind of click over this way and click over that way. I put some golf clubs in a bag here to show you that these heads will have the ability, if I power them with some ATP later, to ratchet like this. And you can guess if I'm connected to something, if I ratchet my heads, I'm going to pull that with me. And that's what's going to happen when we stimulate these myosin proteins, which we'll talk about how we do that at the very end of our story. They act kind of like rowers on a rowing crew. And these oars in the water, the paddle heads here, those would be the heads of the myosin. And they would attach to actin, which I'll just kind of show as being right here floating in the water. There's some actin. If it can, the heads will reach up there and grab the actin and they'll pull it, much like the rowers are being pulled, right? So there's a decent picture of myosin. Now there is a third protein that's barely mentioned, but it's called TITIN, and it is known as the spring. So there is something that has to give elasticity to the muscle, and this is it. So titin is the largest protein in the human body. It's not the largest molecule, that would be DNA, of course, but it is the largest protein that is created from the DNA, and it gives that passive elasticity to skeletal muscles. It also limits the range of muscle. Again, you don't want to over pull the muscle either, so it leads to a passive stiffness. So again, this lets it stretch and recoil. This titin does. It often, if it's drawn in pictures at all, it looks like yellow springs. So here is a very good image. Now you have to remember, first of all, where we are. This thing at the top, this pipe-looking thing, is a myofibril. That's how far down I am. I cannot even see a muscle fiber. It would be composed of many of these altogether. And down below it here, this picture here is showing the same thing, but kind of on a flat 2D kind of look instead of the 3D look that's more realistic up top. So you are looking at a sarcomere. A sarcomere would run from this spot here over to this spot here. There's a length of one sarcomere. And then another one would appear and another one would appear. So the first thing we need to find usually is called a Z-line, or because this is a pipe-like object, if I were to cut it across, kind of like a transverse cut, I would see a disc. So sometimes it's called a Z-line, sometimes it's called a Z-disc, depending on how you see it. But it is right here on this image. I'll just put a little Z for Z-line. Here's another one on the other side, right? Z-line, Z-disc. And then right here in this image, I'll just kind of draw a red line straight down this zigzaggedy line, which you might figure out why it's called Z now. And there is the Z line. Same thing over here. I can come on the right side of this pipe and I see a Z line here, bounded by those two red lines. There's another Z line right there. Okay, that's easy enough to spot. That's the boundary of the sarcomere. Those are the parts that when we do contract this muscle, one Z line will be pulled toward the other Z line, and we will shorten the length of the sarcomere. Let's talk about these other regions. Okay, if I were to just change my color to get some difference here, right here, bounding the Z line, right? So the Z line's right in the middle of what's called the I band. And I can draw it right here too. I can even outline it right there. There is what we would call the I band. In this picture, I can show part of it here. All this, and it would head over to where we find these red guys, these myosins again. So this is the I band. And I can do the same thing here where this myosin ends. I can say all this out here, this direction to the next one would be the I band. Now it's called I band because it's light band. And it's light because it only has one of those sliding filaments. It has actin, not myosin, but it does have the titin. So the Z line and the I band are formed from actin and titin. There is no myosin inside the I band. Therefore it stains lighter, and we call it the light band. The rest of that length is the A band, again called A because it is the dark band. Let me change to a blue here. The dark band has both sliding filaments, actin and myosin. So basically go from one eye to another, and you have an A band right like that. I can start another A band going over this way, and I can start another A band going over this way where I have the word myofibril written. And you can see this A-I pattern, light, dark, light, dark, light, dark. And this is what gives the striated look to skeletal muscle and cardiac muscle. Okay, now the H zone is not discussed too often, but it is the part where we only have myosin. So right in here, I'll call this the H zone. Again, it just usually talks about in textbooks that that's the place where there is myosin only. And then right in the center of that H zone is an M line where the myosin is attached to the rest of the structures. So here's the M line right here. If you want to see it in this picture up here, you can barely see, let's see where I'm drawing right there. Here would be the H zone and the M line right in between it. It's not a Z line this time. That's the H zone and the M line. So they're the major characteristics of a sarcomere. Now to end this talk, we need to talk about a muscle triad. Notice again where we are. I can see a lot of, there's a myofibril, there's a myofibril, no problem. So this must be the boundary of a fiber, sorry. So that would be the endomysium, or is that the sarcolemma? That's always the question. Because it looks like it's the closest layer, I'm going to go ahead and call that the sarcolemma, the cell membrane of the muscle fiber. These guys here should be familiar if you went through the organelles talk with me. Those are the mitochondria. And there's a lot of them stuffed in there, supplying a lot of ATP energy to allow this thing to work. Now, the point of the slide, though, once we found out where we are, is to talk about a muscle triad, implying there will be three objects. And if I change to, say, a yellow color, I can outline that right here. See how there are three pipes there? Right here also there are three pipes. That's going to make up the triad. So we should be seeing, in a good picture anyway, the middle pipe is called a T-tube, or a transverse tubule. Again, I'm going to draw my line right down the middle of a T-tube, and I'm going to bounce it around this one too. So wrapping around the myofibrils, we find these T-tubes. Now, they are extensions of the cell membrane. Remember, the sarcolemma is out here. Those T-tubes, even though they're not shown, they will connect with the sarcolemma at some point. And notice where they're diving down. They're not diving down on the Z-line, because you can see the Z-line kind of here, offset from it. They're diving down right here at this A-I boundary, right? Here's the dark A band. Here's the I band. Here's an I band with the Z line in the center. So we see at each AI junction these T-tubes dive down from the cell membrane, from the sarcolemma, deep around every myofibril. Now off to the side of it, right, if that is a T-tube, we see two other tubes flanking it that are usually a little bit larger, but not always. These two things on either side of it, if that's the T-tube, then these are called cisternae, plural for cistern or cisterna, which is a well. And so if you have the T-tube flanked by these two cisternae, that is called a triad. Now the role of the cisternae is when it is stimulated by something we'll mention in a further talk, it will dump calcium all throughout the cell, especially into the network of tubes that connect those triads. And I can circle that right here. This kind of blue looks like a web connecting those T-tubes and cisternae. That's called the sarcoplasmic reticulum, or the SR. Similar to an endoplasmic reticulum in other cells, but this is specially designed to release calcium. And ultimately that calcium will be released and it will cause the actin and myosin to slide over each other, which we'll see in a talk just down the road. Okay, a lot of details in that one, but very important information. Hope you enjoyed listening to it. Certainly thanks for watching that complicated one. Check out some other videos if you enjoyed it. See you for the next one. Bye-bye.